సుధాకర్ ఇప్పుడు సెట్ అవుతుంది అయింది. వాళ్ళు ఎంతవరకు వచ్చారు చూడండి. కనెక్షన్ ఇస్తా అన్నాడు ఆయన కుర్రాడు. కానీ చెప్తా రెడీగా చెప్పేయచ్చు. సెట్ అయింది. ఫస్ట్ సైడ్ పెట్టి ఉంచాను.
Of SRCP, and today's guest speaker, KPI Professor Producer. Assistant Professor, Department of Pharmacology, Sreenivasarao College of Pharmacy. Representing you for this day 2 session of 1 week of faculty student development program by pharma and healthcare professionals. Academic plus corporate. For this day 2 session, today I am sharing the screen with Professor because to read the formal instruction about and
ఐడి కనపడుతుంది. ఫోటో పక్కన ఉందా?
pleasure immense honor for me in one of the most senior speakers in the pharmaceutical.
Uncle ఎందులో కూడా చూసుకుంటే ఎలా వస్తుంది?
Professor PhD the University
పైన నెంబర్కి ఫోన్ చేసి చెప్పు. సౌండ్ సరిగ్గా వినపడలేదు. అట్లా వినపడలేదు.
At various positions as lecturer, reader, professor, head of the department, and as after his retirement from Anta University in 2010, in 2010, he has been guiding the Board of Studies of 20 He has been the advisor for KV. But Professor Chaudhary sir
Hello.
Numerous such as member of Southern Regional Committee, Chennai and Hyderabad, member of NPA New Delhi, Inspector of Pharmacy Council of India, Chairman and member of Board of Studies of University Pharmacy Departments, National Convener of IPCA,
సౌండ్ రావట్లేదండి. బాగా అవుతుంది కానీ అక్కడ మొత్తం నేను వేరే పక్కన రూమ్ లో మొబైల్ కూడా ఆన్ చేశారు. సౌండ్ రావట్లేదు. మధ్యలో బాగా బ్రేక్స్ వస్తున్నాయి. ఆవిడ చెప్తున్నారు కానీ మేడం గారు అసలు అది అందుకని ముందు చూడండి ఒకసారి. మళ్ళీ విని వాళ్ళకి కూడా నాకు అసలు కనపడలేదు. సౌండ్ వస్తున్నాయి బాగా. అది ఒకసారి వాళ్ళని చెక్ చేసి మళ్ళీ స్టార్ట్ చేయమండి. ప్రాబ్లం లేదు. అసలు ఏం తెలియలేదు. ఇప్పుడు ఏం చెప్పింది నేను పక్కన మొబైల్లో ఫాలో అవుతున్నా. అది
Professor Chaudhary sir has a total of 446 research publications. (171) research papers in national journals and 240 research papers in international journals. Professor Chaudhary sir published 2 books, presented 72 research papers, and delivered more than 70 lectures as chairman and resource person at national. He carried important work within college. Chaudhary sir was the pharmacy 2017 Association Biotechnology and Pharmacy. Today we are blessed and fortunate to have his humble presence here, and it is really a great privilege for me to present you, sir. And thank you for our invitation on behalf of our main faculty, and I want to cordially welcome you.
సెట్ అయిపోయింది పర్లా.
That is unbelievable too. 2010.
హలో,
అలాగే అసలు వాయిస్ ఏం వినబడతలేదు, సౌండ్ వచ్చేస్తుంది. అక్కడ ఏమైనా అడ్జస్ట్మెంట్ చూడండి చూడండి. ఓకే ఓకే ఓకే.
Hello? Hello?
Numerous
Hello?
member of
హలో.
Southern Regional Committee, Chennai and Hyderabad, member of NBA Inspector of Pharmacy Council of India, holder and member of board studies of University Pharmacy Departments, National
హలో.
హలో, అది అమ్మాయి ఇంటర్వ్యూ చేస్తున్నారు. ఇంకాను డిస్టర్బెన్స్ స్టార్ట్ చేసుకోవచ్చా నేను? ఓకే 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 ఓకే. అంటే నాకు ఇక్కడ వాయిస్ ఏమి వినబడతలేదా తను చెప్పేది? హలో, ఒకసారి మళ్ళీ కొద్ది ఏదో ఏంటంటే బ్రీఫ్గా చెప్పండి. అమ్మాయిని వినబడుతుందో చూస్తాను. అది వాయిస్ వినబడతలేదండి. నేను ఇక్కడ అవును కరెక్టే, కానీ అసలు తర్వాత వినపడాలి కదా, అదేమి వినపడతలేదు. థ్యాంక్ యూ.
Convener of IPCA
సౌండ్ రావట్లేదండి, బాగా అవుతుంది కానీ అక్కడ మొత్తం నేను వేరే పక్క రూమ్ లో మొబైల్ కూడా ఆన్ చేశారు, సౌండ్ రావట్లేదు, మధ్యలో బాగా బ్రేక్స్ వస్తున్నాయి. అవి చెప్తున్నారు నేను మేడం గారు, అసలు అది ముందు చూడండి మళ్ళీ వినియోగం కూడా నాకు సౌండ్ బాగా అది ఒకసారి మనం చెక్ చేసి మళ్ళీ స్టార్ట్ చేయమంటారు, ప్రాబ్లం లేదు. పక్కన
వాల్యూమ్ కంట్రోల్ చేసుకోవచ్చు.
Shall I start? Dear Dr. Sudhakar, hello, shall I start? Sound is it anybody can clarify? My voice is clear? Hello? Hello, Dr. Sudhakar? Hello? హలో హలో కాదు, వాళ్ళు ఏదో మరి చెప్తుంది కదా అమ్మాయి, ఓకే సరే, నేను నా వాయిస్ సో ఉందా, వినబడుతుందా? ఓకే 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 ఓకే, థ్యాంక్ యూ. Dear Dr. Sudhakar, Principal of Sreenivasarao College of Pharmacy, and his team members and learned faculty members and prospective students of pharmacy of various pharmacy colleges. Good morning to all. I extend a warm welcome to you all to this session. And initially, I thank the organizers, particularly the principal, Dr. Sudhakar, and the IPA, AP State Branch, the President, Professor P. Rajeswara, for providing me this opportunity to interact with you all through this online faculty development program. The topic of this session is role of biopharmaceutics in drug development, and it covers an overview of the drug development and approval process, different stages involved, and the role of biopharmaceutics, the essential concepts of biopharmaceutics and their application in the drug development process at different stages like the drug design, dosage form development, commercial technologies involved, and evaluation methods, particularly dissolution method development, and conducting the bioequivalence testing, and such other aspects will be covered in this presentation. Drug development and approval process. So all of you know that the objective is to develop safe, effective, and quality drugs and drug products and devices for human use. And this is a very challenging research aspect and always innovative. And the industry normally concentrate on this innovative area of research. And since long, this is one of the important area of research in pharmacy, the drug development and approval process. And it is carried out in different stages, and this requires expertise in different disciplines like the organic chemistry, particularly synthetic organic chemistry, biotechnology, and phytochemistry and herbal drugs, etc. Such knowledge. So this is because the first stage in the drug development process is known as the NCE development. The NCE, new chemical entity development. And since long, the major source of this NCE is the organic synthesis. Even today, it is the major source for developing drugs, newer drugs. And several thousands and thousands drugs are designed over years, and they are all in the practice today. And still there is a lot of demand. Continuously, there is a demand to develop newer and newer drugs because of the changes in the disease pattern, and newer diseases are coming up in the community. With the result, the development of drugs is always a continuous process. And another source of this new drugs is the biotechnology. During the last 3 decades, several life-saving, wonderful medicines have been developed using the microorganisms and other living sources, employing the technology under the biotechnology component, and excellent growth has come in this area, and wonderful drugs are derived by biotechnological means. So essentially, the drugs are of 2 types. One is those developed by synthesis, they are known as the chemical drugs, and the other is from natural sources, particularly the protein and peptide drugs. They come under the second category called the biological. And they are different in their character, in their properties, and how they react in the body. In several other aspects, they are entirely different. The major difference is the chemical drugs are more stable, whereas the biologicals, which are the proteinous, protein and peptide based drugs, enzyme based drugs. These are very sensitive to the environment, and hence special care has to be taken in their development, in their manufacturing and evaluation and such other steps. And during usage also, these biologicals, they produce a lot of reactions in the body. This is because they are immunogenic. Whereas the chemical drugs are totally non-immunogenic and safely we can use it. But whereas these biologicals, their usage is rather difficult. And today, all of you know that the people are giving more weightage for pharmacovigilance. And this is all because in the recent past 20, 30 years, more and more biologicals have come. And though they are approved as the safest drugs during the earlier phases, all the phases of the development, but after usage also, you may find unwanted adverse reactions, and hence it's a continuous process to record the adverse reactions that are going to be there while using. Like the two sources, they are widely used today. Along with them, since long, there is another very important source of drugs, that is phytochemicals or plants and herbal drugs. So since long, this is a source for wonderful drugs, but over years their use has declined. The reason being that there is no proper evidence, scientific evidence for their claimed effects. And today, the public, rather the patients, they are well aware of things. And what is now needed is evidence. So evidence-based medicine. That means now today, not only the doctor, the patient is also asking for evidence. What guarantee, what evidence the doctor and pharmacist can give that the particular drug will be curing my disease. And several of the wonderful hand drugs are derived from nature. They are safe and they are useful for the disease indicated, but there is no evidence. Evidence is missing. And hence now the efforts are going on to create evidence for the scientific evidence for their claimed properties. When once it is developed fully, and these drugs will be accepted in total. Now you see that presently we are in the middle of a big problem. But all of you know that the COVID-19 pandemic. And earlier, if you go back to 6 to 7 months back, this COVID is not known. And we are not worried, lived happily. Now during the last 6 months, now the entire world is worrying about this COVID-19 and how to get rid of this. What is the cure? And so far there is no cure, and now it is a scientists are struggling hard to develop the cure. Probably because it's a viral infection, the best cure will be a vaccine. And India is strong in the biotechnology, particularly in the vaccine manufacturing, and globally, 60 to 70% of the global demand of vaccines will be supplied is supplied by India only, and hence the entire world is looking at India for a vaccine to be developed and supplied to save the humanity. And this shows that the research under this title, that is development and approval of drugs, safe drugs, is a continuous process. And is a continuous process, and there is always a demand for this research. And then when once the NCE is developed, directly it cannot be put to use in the humans. And then the next stage is that NCE characterization. Several properties of NCE, like the physical properties, chemical stability characters, and biological properties, pharmacokinetic properties, all these properties are to be assessed. Some in the laboratory, characterization by chemical tests, by spectral studies, etc. And some require the testing in animals, like experimental animals, for assessing the pharmacokinetic concepts, pharmacokinetic parameters, as well as for the pharmacological effects, may be beneficial or otherwise, but thoroughly investigated the various possible effects on the body, including the toxicity. And hence, in the NCA characterization, you find different types of evaluations. It is also a type of research. And this research is of two types. One is the preclinical testing for efficacy and toxicity or safety. So with regard to this research, the much weightage is given not only to efficacy but to the toxicity also. The drug developer should be safe and then it should be effective. And another important third quality is it should be a good quality. Then only the drug will be accepted and it can be put to use in humans. And for this purpose, the experiments are to be conducted as per the science, as per the ethical practices. And every country is having a drug regulatory body organization and which is entrusted with the responsibility of controlling the way in which the testing is being done right from the beginning, and certain guidelines in the form of GLP for the laboratory investigations and preclinical investigations, and in the form of GCP, good clinical practices for all the clinical trials to be conducted, and in general, certain other guidelines regarding how the research is being done is provided even in the GMP also. And hence, the regulating bodies globally, they are giving certain guidelines like this, and it is very necessary that the scientists who are involved in this research, development research, should necessarily follow them to ensure that the data collected is genuine, is free from bias, and adequate. And with the result, it can be used safely to make certain conclusions and evaluation of the effects, etc. And next is the pre-formulation and formulation development. So drug is, of course, all of you know that is used in the form of a delivery system or a dosage form. So certain procedures and practices are to be followed in the formulation. Before formulation, the properties are to be thoroughly evaluated, that is otherwise called the pre-formulation and formulation development, and newer and newer types of delivery systems are being developed, and it is now an interesting area for this drug development and approval process. And the formulation development is really challenging one, and which gives long life to the drug and also several benefits to the patient, and hence is another important component of the process known as the drug development. And then so far the amount of work done under various headings, this is all the stage is known as IND, investigational new drug application, at the end of which the entire data is compiled in the form of a dossier and then submitted to the regulating bodies. They make a thorough evaluation, and in the IND, the sponsor company or the researcher requests the regulating body to give them permission to go for clinical studies. Clinical means evaluation in human subjects, that is ultimately needed. And so the clinical studies will now come into, and they're done in different stages like phase 0, phase 1, phase 2, 3, 4, etc. And then these studies, the emphasis is given to both efficacy and toxicity. Initially in the phase 0 studies, or initially with a small group of subjects, human subjects, under controlled conditions, the assessment is done, particularly for fixing the dose and for enlarging the dose and for assessing the parameters of ADME and following the therapeutic effect, developing the quantitative measures for therapeutic effect as far as possible, and assessing the route of administration and fixing the route probably. Initially, all these aspects will be evaluated in the experimental animals, but certainly in the phase 0, again they are done in humans to ascertain how far they are applicable to human subjects. When once it is satisfactory, then the phase 1 studies will start under the supervision. All these clinical studies are to be done under the supervision of a competent doctor, or doctor also forms a part in the drug development process, and under their guidance and suggestions, the evaluations are being done in a small number first in the phase 1, observed especially for the efficacy and toxicity. Then the number of subjects will be going on increasing in the phase 2, and again all observations will be made. And phase 3, again the number is large, enormously increased to 3,000 or even more, 5,000 or even more. And the testing is spread in different parts to ensure that the result is applicable to all the humans in different countries, different races, etc. And so it is a vast testing is involved. And after completing all these studies, again the sponsor company has to submit this to the regulating body in the form of NDA, new drug application, and again ANDA, abbreviated new drug application. This is particularly applicable in the case of generic drug formulations, etc. And the assessment is made, again a permission will be given for marketing. So this is also known as the marketing authorization. Then once the marketing authorization is there, and essentially the rules remain almost the same, but little variation in the different countries, and hence in the country in which the sponsor want to market the product, he has to follow the guidelines and work to their expectations and obtain the data as to meet their requirements, and then you will get the approval. And once the product is approved, then again during usage the drugs are followed. This type of is known as phase 4 or post-marketing surveillance. Now the word pharmacovigilance is given to this type of following, and the pharmacist, the doctor or the pharmacist, and together in hospital while it is in usage, the reactions, the possible reactions are to be followed and assessed and reported properly in the desired manner. Then only the regulating bodies, after assessing periodically, will take a decision whether to continue the drug in the market or to withdraw the drug. And several decisions will be taken by the regulating body and the concerned body. And hence, in the entire the drug development and approval process, the regulating bodies have great role in checking the quality right from the beginning. And for that purpose, at stages like IND, NDA or ANDA, and afterwards through pharmacovigilance, etc., thorough check is made not only on the drug, but here it is a research, and hence the research, how well it is being conducted in the organized way, and guidelines are given, and hence the data integrity should be there, and the regulating bodies, they check the validity of the data and then sanction the approvals or otherwise. And pharmacovigilance is, of course, is a continuous process. And hence this is an overview of the drug development and approval process. And suppose this indicates it's becoming an increasingly challenging task, and a multidisciplinary research. Several subject experts are needed. Knowledge and skills in several subjects like organic synthesis, microbiology, biotechnology, analytical techniques, pharmacological evaluations, and toxicological testing, formulation testing, formulation development, manufacturing, process development. So, like this, several scientists of different disciplines, they are integrated their expertise and contribute to the development of the new drug. And hence, it's not only the pharmacist is involved, along with pharmacists, several scientists are needed to develop it. And pharmacy, as you, as you know, again in the course itself, it is a multidisciplinary, include almost all of the knowledge that is needed, and hence the pharmacist is the right person thoroughly involved in the research needed for drug development and approval. It's a lengthy process, as seen, nearly estimated that 10 to 15 years is required to develop a new drug. And now, with relation to the COVID-19, okay, it requires time. And the public, not only why public, even the doctors, without knowing fully the process involved, may say that in 1 month, in 2 months, the drug is coming out. This is all, maybe all these drugs are for temporary measures, but approvals will not be given. And a few days back, one organization it's claimed that they developed a drug for COVID, but immediately objection has come from the government quarters that there are no approvals for such a drug. So it is now the pharmacy teachers and students should understand that the implications involved in drug development discovery process, it is definitely a lengthy process unless all the tests are being conducted and approved, it is not possible to market. So the immediate requirement is research has to be augmented in the required direction. Especially, we are looking at a vaccine, and hence the biotechnology should concentrate their efforts. And I wish that soon they may succeed in developing a vaccine. But evaluation process will continue, which may require, if not 10, 15 years, because already technology is fully available. And if not 10, 15 years, at least a minimum period of 2 years, etc., is needed to come up with a good approved vaccine product. Similarly, it is very costly. It's estimated anywhere between 800 millions to 1 billion US dollars is the total cost, and that is the reason why only the multinational big companies are engaged in such type of research, and even then they cannot alone, a company alone cannot do all the type of work, and hence the work will be assigned to other groups. These groups which are capable of doing research needed, they are known as the contract research organization, CROs. Excellent growth observed, and now in fact the India has become the center for drug research, not only for drug manufacturing, but drug research also. India has become the center. Many pharma industries, along with several CROs, established and are doing good business. Even then, we are lacking in certain areas. We are excellent in vaccine development and manufacturing. We are excellent in herbal drug products. We are excellent in the manufacturing, particularly generic products, and exporting to almost all the countries. But we are lagging behind in certain areas like the synthesis development, drug development itself, basic bulk drug development itself, we are lagging behind. Still, we depend upon a large, other countries getting the drugs. But I think all the pharma industries in India, they are getting either the raw material in the form of an intermediate or an NCE or a bulk drug API from China only. And now you see that, and the Chinese, how wise it is to accept the Chinese products and Chinese technologies is doubtful. And hence, we need to develop in the areas where we are lagging behind, particularly in the making the API development and manufacturing. And in the same manner, this is suppose to achieve one molecule, one NCE, it requires to assess, to study nearly more than 5,000 molecules, and out of which only one may turn out to be the good one. And also a large number of subjects in the clinical stage, clinical trial stage, as many as 5,000 or more subjects are involved, and then stringent regulatory control is there. It is needed, but it is also a challenge to meet their requirements. The research has to be carried out in that direction and develop new drugs. And so, like this, it is a very essential component. Now, coming to this biopharmaceutics, essentially, I'm going to talk now about what is this biopharmaceutics and how important it is in the drug development process that so far we consider. And the biopharmaceutics, simply you can break into two words. One is bio and the other is pharmaceutics. See, bio here refers to biological system to which a drug is supposed to be administered. And pharmaceutics, all of you know that it is science that deals with the development of or formulation of dosage forms, manufacturing, testing of dosage forms, etc. And hence combine these two. Why you need to combine is it is well clearly established now that the formulation properties, okay, related to drug, related to dosage form, significantly influence therapeutic response that is observed after its administration. And hence, it is the science that deals with the correlation or relationship between the properties of the drug and dosage form and the therapeutic responses observed following their administration. And hence, to fit into this concept, the biopharmaceutics include several things. One is studying the drug absorption, distribution, metabolism, and excretion, that is otherwise called ADME. It is a study of ADME and the process involved, factors involved, mechanisms involved, etc. And then the study of the kinetics of ADME. So kinetics of ADME is very important because all these processes are rate process. The drug is administered to the system. It's not staying permanently in the body. It is a guest molecule to the body, and hence it is received, it is handled, and then it is eliminated from the body, ADME. So the rate at which each one is occurring, the kinetics are very important. That determines the therapeutic effect observed or not, the toxic effect, is there any possibility, etc. All these depends upon the kinetics of ADME only. And the kinetics of this ADME is otherwise known as the pharmacokinetics. And hence the pharmacokinetics is an integral part of biopharmaceutics, and together always go, and both have wonderful applications in drug development. And in the same manner, this also includes study of the relationships between the, otherwise you can put the drug. It has certain properties, certainly physical properties are there, chemical properties, biological properties, kinetic properties of drugs, and the therapeutic effect. So on one side we have all the properties, on the other side we have the therapeutic efficacy. And hence the therapeutic efficacy is measured in terms of three important characters. One is onset of action, and another is intensity of the effect, and the third one is duration of the effect. So all these three important characters of the therapeutic effect are being altered by several of the properties under the headings like physical, chemical, biological, and pharmacokinetic properties of the drug, and also then comes the formulation, that is the dosage form, type of the dosage form, ingredients in the dosage form, how it is manufactured. All these dosage form related factors will also influence therapeutic response. And then this application of the knowledge in drug development process that is ultimately needed. So a part of this subject, biopharmaceutics, is learning the principles, and the other essential part is that application part, how well you can apply the principles studied in the different stages in drug development process, etc. And hence, one is learning the principles, and the other is applying it in the area where it is useful. Okay. Now, areas of application of biopharmaceutics. There are several areas where it can be applied. The first part is in the drug design. The drug design means development of a new chemical entity, NCE. So to increase the efficacy of the NCE may or may not have the required properties to produce any effect, and hence by altering the chemical configuration or chemical modifications are done continuously, with the result to achieve certain characters that we see later, and hence the knowledge of biopharmaceutics is very important in the drug design, always to make the drug rapidly absorbable and to be distributed uniformly without any accumulation, etc., and be made available to the site of action, and ultimately the drug has to be removed from the body. All these are important, and hence the by modification, chemical modification, and these characters can be altered, and where the knowledge of biopharmaceutics is very much useful. And next is the selection of the route of administration. Route. So the route of administration. There are several routes of administration, and which route is suitable for a given drug, NC, is a new drug, etc. This has to be done, evaluated in experimental animals as well as in the phase 0 studies in the clinical setting. And so there again, the principles of biopharmaceutics are applicable. And now the determination of the dose and dosage schedule. This is again an important task. And for a new drug, how the dose is determined, and for a known drug also, how the dosage modifications are made and how the dosage schedules are designed. This is all an important application of this biopharmaceutics through the pharmacokinetics. And the pre-formulation studies are very much needed before formulation to assess all the required properties is also a part in the biopharmaceutics. And then development of dosage form or delivery systems based upon the principles of biopharmaceutics will be well applied in this area. And then similarly, evaluation of bioavailability and bioequivalence for comparisons, etc. This is another area. And evaluation of drug-drug interactions, drug-food interactions, and similarly therapeutic drug monitoring, and the dosage adjustment in situations of altered physiology or clinically in the patient suffering with certain diseases. Then similarly, ADME, basic understanding about the ADME of a drug. Such studies also are very important to understand the mechanism of action of the drug and efficacy of the drug, etc. And later on, we concentrate on some important concepts called the principles of biopharmaceutics. Principles of biopharmaceutics. So here it is, principle 1. So this is the first one is the therapeutic response of the drug depends on its concentration at the site of action in the body. So it is well established today that the therapeutic effect produced by a drug is dependent upon the concentration of the drug at the site of action. Site of action will be in the body in one or other tissue. And hence, though it is ideal there is no way to find out exactly what is the concentration of drug in the tissue where the receptors are present and with which the drug has to interact to produce a therapeutic response. And hence, an alternative to this is the plasma drug concentration, which you can easily measure it precisely, and it is taken as a concentration as a measure of the concentration at the site of action. It is almost equal or it is proportional. The ratio is always a constant. So there is a direct relationship between the plasma drug concentration and the concentration at the site of action, and hence plasma concentration is taken as a measure of the concentration at the site of action, basing upon which the therapeutic performance of the drug and the dosage form are evaluated. There is no other way. And ultimately, the pharmacological response can be measured, but even today, the measurement of pharmacological response is not possible with many drugs, particularly in the clinical setting. And hence, the best way is to assess the performance of drugs and drug treatments is only the plasma concentration. So therapeutic responses, okay, such as onset and intensity and duration of action observed following the administration, now all considered as dependent upon the drug concentration only, plasma drug concentration. And it's not only therapeutic effects, but even the toxic effects are also depends upon the plasma or blood concentration achieved. If the concentration is very low, it may not even be sufficient to produce therapeutic effect. That is known as a subtherapeutic concentration. In certain range of concentration in the plasma, the drug will be producing its effects, therapeutic range. Then above that, it may produce toxic effects, and beyond, still above, it is known as a laser, and hence death may occur. So it is all the effects, whether the desired effects or unwanted effects, toxic effects, lethal effects, it all depends upon the concentration only. And that shows the importance of the blood concentration profiles in this subject. And now the blood level curves are important tools for interpretation of the therapeutic response. Out of this, now we can conclude that the principle 2 is the entire biopharmaceutics and pharmacokinetic developments are based upon their applications are based upon the studying the blood level curves only, and hence the blood level curve is an important tool in this subject. This is a typical blood level curve following the oral administration. The shape of this curve will be different if the route is changed, and this is the typical blood level curve when the drug is given by a route involving a process of absorption, or when the drug is placed outside the central compartment known as the blood, because this is the blood concentration is followed. If the drug is given by intravenous injection, the starting point would be somewhere above the peak and somewhere a higher point on the y-axis, and later on the decline will be similar to the decline phase in this curve. And hence the route of administration will alter the blood level curve. But many times the oral route is given preferred because of various reasons which you are familiar, and this is the typical curve. So the concentration will be going on increasing, reaching a peak, and then going on decreasing because of the elimination. In the first phase, known as absorption phase, the concentration will be going on increasing, and as soon as the drug reaches the general body circulation, the body will try to eliminate, and hence there will be an elimination phase also, but not evident in the beginning, because in the beginning up to peak time, the absorption rate is greater than the elimination rate, and hence the build-up of concentrations will occur and reaches a peak value, then decline, in which the absorption rate
decreases and after some time it ceases, there remains only the elimination phase continuously until the drug is totally eliminated. And you see that there is a minimum MEC, minimum effective concentration, and similarly minimum safe concentration or minimum toxic concentration. So in between it is called the therapeutic range. And originally the concept of minimum MEC, minimum effective concentration, was originally developed with the antimicrobial substances that do during the Second World War time, when there is a lot of demand for developing antimicrobials. Several microbials were developed, but how to assess it? They realized that a antimicrobial agent should present a minimum at a concentration. Minimum some concentration is needed to be effective, otherwise it is not effective. So coined the word minimum effective concentration with antibiotics. Later on over years, the same concept was found true with almost all types of drugs, and now for every drug there is a minimum concentration needed in the body to be effective, known as the minimum effective concentration. Similarly, the other concentrations also, this one information you have to generate in the drug development process, as well as all the information should be made available. Now wonderful books are there related to therapeutics and pharmacology. All the data for a old drug, for a known drug is available. For a new drug, somebody has to carry out investigations first in the experimental animals, then in the clinicals study, and to establish all these concentration effects, etc. So this is the way in which you can interpret. So following the concentration, okay, onset of action is the time at which the concentration crosses the MEC. That means action is initiated. And as long as the concentration is within therapeutic range, effect sustained, that is known as duration. And later on, the concentration when falls below again MEC due to elimination, the effect is lost. And before that concentration declines, normally in the dosage schedule you will give a second dose, and like that a third dose. And that is the reason why the drugs are normally used as per a schedule. So this schedule also has to be developed based upon the pharmacokinetics only. And then these blood level curves, okay, so several factors will influence these blood level curves. One is the route and the next is dose. The more the dose, the more will be the concentration. The other things being equal. And hence the drugs are to be used at a particular dose, and you have to develop that dose first in the animals, then in the human subjects. This work is done in the clinical phase 0 and phase 1 studies sometimes to finalize the dose. And dose will vary from the route also based upon the route. And dosage adjustments are certainly needed when they are put to practice in the clinical practice. Then dosage alterations for that, it is based upon the pharmacokinetic characters. Then the absorption rate and extent. One of the important factor or concept, absorption rate, completeness of absorption. To what extent the dose given is absorbing, and rate. These two will determine the shape of the curve and form an important factor. And the physical properties, like all are important, like aqueous solubility, lipophilicity, partition coefficient, permeability, molecular size, particle size, form, means crystalline or amorphous, and polymorphism. All they appear to be simple, but they are very, very significant, influencing the therapeutic efficacy. If you still remember the way in which the book in the medicinal chemistry books and pharmacology books, the topic is discussed sometimes as the crystalline medicine, crystalline forms are devoid of therapeutic efficacy. That is even before the emergence of biopharmaceutics as a distinctive body of knowledge. Earlier also, people noticed that the crystalline drug, they do not produce any effect. That is Okay, the observation recorded since long. So why then the answer comes when the biopharmaceutical? Why crystalline forms are not producing any effect? Yes, because the crystalline forms they dissolve very slowly. They dissolve very slowly, and dissolution is very slow. Simultaneously, the absorption also is very slow, and hence in the body the drug absorbing slowly, and whatever is absorbed, it is being rapidly eliminated by the process of elimination, with the result at no time the desired concentration will generate, and hence the drug they do not produce. This is after when the subject of biopharmaceutics is fully developed and understood. So the answer is like this is the reason why the crystalline drugs are described as devoid of therapeutic effects. So like this, the amorphous forms is certainly they dissolve fast, they absorb fast and produce the effects in a good manner. Like this, almost all factors will influence polymorphism. Now, making changes in these properties is an important task in drug design. Making these properties is an important task in a formulation or as well as in the drug design. Some of these things later on we'll see. All are important. Again, at different stages we will be highlighting each one. Then the chemical properties of the drug, like the pKa and degree of ionization, that will influence the absorption and hence the shape of the curve, and the chemical form, whether it is salt form or parent compound, stability, all these chemical characters also influence, and biological, the pH in the tract or at the site of administration, and gastric emptying in the case of oral, and first pass effect again. All these the biological properties, some of them, and the pharmacokinetic like half-life, clearance, and excretion rate. These are all the parameters related to the elimination of the drug. Then volume of distribution and localization, binding. These are the factors related to distribution, and like this. So drug-drug interactions, drug-food interactions, all of them will alter the curve, blood level curve. Now, if it is altered, what problem? Yes, if it is altered means the characters will change, and hence the therapeutic responses, properties can greatly be altered by manipulating one or more of these all these characters. Principle 3. So in the biopharmaceutics, normally, okay, when the drug is administered, the drug has to cross certain barriers, biological barriers, at every stage. During the process of absorption, it has to cross, and during the process of distribution, again, excretion, it should cross the biological barriers, and hence, in all these processes, the passive diffusion is the major mechanism. Passive diffusion, and so it obeys as per the passive diffusion rules. And so the majorly passive diffusion. Of course, there are other mechanisms also to some extent they influence the process like active transport, but majorly passive diffusion. And then the principle 4. Lipid soluble molecules cross the biological barriers rapidly and readily. So lipid. Why this reason is that in the body, all the biologicals they are lipid in nature, more lipid in nature, biological membranes, more lipid barriers only. Whereas the fluids that are included in the biological membranes are inside biological membranes, surrounded by biological membranes, all the fluids like tissue fluids, like the gastrointestinal tract fluids, secretions, all are aqueous in character. Okay, so two different characters. The fluids are aqueous in character, membranes are lipid in character, and hence for diffusion, the lipid soluble molecules can readily cross the barrier and enter into all the process will occur rapidly. So lipophilicity and high partition coefficient otherwise. Lipophilicity is a character, okay, which refers to the solubility in lipids. And similarly, high partition coefficient, how the lipophilicity is assessed is by determining the partition coefficient P over W against water and oil. So oil by water, if you define, if this partition coefficient is reported, and this is reported as P value or log P value in all the literature, based upon that, you can make certain inferences about the lipophilicity. So lipophilicity, okay, and high partition coefficient towards oil facilitate rapid absorption and distribution of the drug. Okay, there are certain ranges identified as if the log P is equal to 0 means it's a balanced one, hydrophilic and lipophilic character is balanced and it's rapidly absorbed. The best example is alcohol. Alcohol has equal miscibility in water and alcohol and oil, and it very rapidly absorbed even orally. Okay, because P is log P is 0, and minus log P indicate it is more hydrophilic, and plus log P indicate that it is more lipophilic. Lipophilic, and hence the positive log P indicates the lipophilicity. So log P in the range of 0 to 3 is ideal. 0 to 3 is ideal. And hence in the drug design, it's not that always the NCE that is developed initially is fulfilling this requirement. To be effective and to exert its effect biologically maximum, to find a place in medicine, in the therapeutic use. So this adjustment has to be further made. Measure the simple parameter log P is measured, and if it is in this range, to take it granted, it can be used, and other manipulations can be done to still improve, if needed, the absorption, penetration character, otherwise, etc. And suppose if the P is not in this range, now you have to think about how to modify. And many times the lipophilic character has to be created into the molecule. Lipophilic character has to be induced or created. And hence, organic drugs are sufficiently lipophilic. Organic drugs are sufficiently lipophilic and cross the biological barriers readily. Easily you can do it. And today, several of the drugs available for use in the practice, nearly 95 to 98% of the drugs are organic drugs only, and hence they are sufficiently lipophilic. In their design, chemical design development itself, the lipophilic character is there or introduced into the molecule, and by the time it is available, they are sufficiently lipophilic and hence cross the biological barriers readily. And increase in lipophilicity, okay, increase the absorption and distribution of the drug. That is one general principle. In drug design, lipophilicity is induced or increased by certain manipulations, chemical manipulations, like increasing the carbon chain length. If you go on increasing the carbon chain length, okay, the lipophilicity will certainly increase, but there is a limit. You cannot go on increasing much. So up to 5 carbons in the chain, it's okay. Otherwise, if you increase beyond that, now it loses another important character, that is water solubility. Water solubility. So equally important is the water solubility, aqueous solubility. That will come later. And hence, by manipulating the chain length, also it is possible to alter the permeability or partitioning. And then by replacing certain of the polar groups, OH groups, by the alkoxyl groups, such as many times methoxyl derivatives are made. This is to improve the lipophilicity, and such other things will be done by preparing esters and such other derivatives which are more lipophilic. It is possible to alter is possible to alter the lipophilicity, and hence in the design itself, the required extent of lipophilicity has to be introduced into the molecule for its better use. The principle 6 is aqueous solubility. It's also essential and another important factor for drug absorption. And the compounds which are not soluble, insolubles, okay, again they do not exhibit the therapeutic efficacy. Similar to crystalline drugs, here also the compound having a low solubility, very low solubility, aqueous solubility. Solubility means always aqueous solubility unless otherwise specified. So it is one of the important factors, solubility, and the principle is that in biopharmaceutics, one of the important concept is that molecules from solution can only cross the biological barriers. That means now, if you want to understand how the drugs absorb or cross biological barriers, the particles cannot cross the barrier. It is only the molecules. Molecules means it is in a solution form. So when a drug dissolves in a solvent water, it forms the molecules. And these molecules from solution, they can easily cross the biological, provided the molecules are sufficiently lipophilic. And hence organic molecules are all sufficiently lipophilic and hence readily cross. If given in the molecular form, otherwise means if they are in the soluble form. But the real problem is that the organics, because of their organic nature, they are not soluble in water. They are having very limited solubility, and hence they again create, though they are permeable, but because of the insolubility or not soluble freely in water, they create a problem in the absorption. They are considered as poorly absorbable again. And so all solids administered shall undergo a process known as the dissolution and form a solution in the aqueous fluids that surrounding the particles. So when you administer a tablet or a capsule or a suspension, whatever, in all these cases, the drug is in the particulate form. These particles cannot cross, so they have to undergo a process called dissolution. So dissolution is a physical process in which drug molecules from the solid surface will be released and enter into molecules in the solution. It is said as dissolving or soluble. So drug molecules from solution then cross the biological barriers and enter into plasma. So we can say that the drug is absorbed. So remember that the particles, whether it is a fine particle or a coarse particle, can never cross the biological barrier. It should undergo a process called dissolution, and hence dissolution is formed an important step in the overall absorption of the drug. And for several organic drugs, the dissolution was found to be the rate limiting step, and why it is becoming a rate limiting step, because all organic drugs inherently they are insoluble in water and dissolving at a very low rate in the surrounding aqueous fluids in the tract, and once they dissolve, then only they can absorb. Without dissolution, they cannot absorb as particles, and hence the dissolution or solubility in aqueous environment is a very important requirement, and all the body fluids are aqueous in character. Then hence the solubility forms another important property in biopharmaceutics, and drugs having low solubility. So it is estimated that when the solubility of a compound is less than 0.1 percent, 1 ml, okay, 0.1 gram per ml, here some word is missing, 0.1 gram per ml, exhibit the problems of invisible insolubility. And hence, one of the important characteristics to be assessed is the aqueous solubility. The aqueous solubility, if it is more than that, it is sufficiently soluble, then you can easily work up into different formulations, etc. But if it is not, then you have to take some measures to increase the solubility. And that's what you need, what happens in the case of all organic molecules. Then the dissolution. Dissolution is the process in which the molecules from a solid surface are transferred into a non-reactive surrounding fluid to form a solution. And dissolution is a prerequisite for absorption. Without dissolution, we cannot expect good absorption for a drug. And dissolution is the rate limiting and is characterized by the rate of dissolution. It's the rate process, and hence it is characterized by the character known as the dissolution rate. And thence the dissolution rate is directly proportional to solubility. There is a direct relationship between the dissolution rate and the solubility. The more the solubility, the more will be the dissolution. And organic drugs exhibit low active solubility and hence low dissolution and hence low absorption. All are low. Dissolution forms the rate limiting step in the absorption of several drugs, and about 95% or more come under this category. Dissolution is the rate limiting step, and dissolution, that is why in the modern books of standards, that is pharmacopoeias, the dissolution rate specification is mentioned for several drugs, almost all tablet and capsule dosage forms, irrespective whether the drug is soluble or insoluble. Even for soluble drugs also sometimes the dissolution forms a critical. The reason is that dissolution not only depends on the properties of the API but also on the formulation ingredients, and hence dissolution of the dosage forms is an important concept, and all pharmacopoeias prescribe dissolution rate test specification for almost all tablet and capsule and such other solid dosage forms. And the concept is found to be very important even in the case of the liquid dosage forms like the emulsions and semi-solid dosage forms like suspensions. Unless the drug undergo dissolution, you cannot expect any absorption and entry into the general circulation. Without entry into the general circulation, you cannot expect any therapeutic effect. Then next principle 12 is biopharmaceutical classification system. This is based on the solubility and permeability, two important characters. So we have seen already solubility and permeability. It's a complex term. It is essential that permeability include the lipophilicity as well as the molecule size. Size is also important, and to some extent the partition coefficient. So all these three together, they are summarized in the word permeability. But permeability normally is taken as an alternative to the lipophilicity, and hence the two important characters are solubility and permeability. So based upon these two characters, all drugs are classified into different categories. This classification system is known as the biopharmaceutical classification system, which forms a basis for understanding the properties and for doing the formulation work. And the class 1 means high solubility and high permeability. Both the properties are high on the higher level, and hence virtually they do not create any problem in the absorption. So even a tablet or a capsule behaves like a solution when given, and virtually there is no problem. So their solubility is not at all an issue, but the absorption depends upon the partitioning or the or the lipid solubility of the drug. Lipid solubility. And just now we have seen that even for soluble drugs, dissolution is very important. For example, these are highly soluble. Diltiazem hydrochloride is a very highly water soluble drug. Even then, in the pharmacopoeia, the dissolution test is there. The reason is the formulation additives that are added in a tablet formulation will significantly increase the dissolution. So, in the case of class 1, some examples, there may be many examples in the literature: metoprolol, diltiazem, verapamil, propranolol, all such type of drugs which are having highly soluble hypermeability, virtually no problem in their formulation. You can easily formulate whatever type of dosage form you need as a liquid or solid or modified system, etc. And class 2, it is a low solubility and high permeability. So inherently, the permeability is good, but they exhibit low solubility. Here it is all organic drugs come into this category. Like we have the drugs like telmisartan, nifedipine, phenidine, dibenclamide, all drugs, several drugs today in usage, they are under this category only because they exhibit low solubility and hence the dissolution is a rate limiting step. So their absorption is dissolution rate limited, and hence you have to take care about the dissolution character in their formulation. And they are well suitable for all types of formulations, for all routes of administration, etc. And class 3, again, this is a high solubility but low permeability. Inherently, permeability is less, but they are having solubility. So permeability has to be increased. The permeability forms the rate limiting step, and several permeation enhancers have been developed in recent years, and lipid-based drug delivery systems have been developed to tackle this issue, and hence some of them we will discuss later on. So this class 3, the permeation is the rate limiting step, and provided the permeability is taken care, they are well suited again for oral absorption, other routes of other dosage forms also we can go ahead. Then drugs like cimetidine and aciclovir and like captopril, all they come under this category. And class 4, that is both are less, low soluble and low permeability, like Taxol, chlorothiazide, comes under this category. Also modern drugs, and their performance is unpredicted given orally, and hence they are not recommended orally, but alternative routes are available, alternative routes, and hence sometimes the parenteral intramuscular route is available, and which also rapidly produce the effect, the desired effect, and such. So non-oral routes are taken up for this type of drugs. So like this, when once during the developmental stage itself, the concerned properties if we evaluate, important is the active solubility and the permeability or lipophilicity in terms of the partition coefficient O/W. If we assess these based upon and the characters, the drug can be classified into one or other of these categories. Basing upon the category, you can try to develop a suitable means, technique, or a dosage form to enhance its absorption and distribution character and to be more effective. And then many drugs are under class 2, and in while defining this class 2 definition is the high permeability and low solubility. But before going into that, so we are using the terms highly soluble or highly permeable. What do you mean by what is high and what is low solubility? Similarly, what is high permeability, low permeability? So it is in the biopharmaceutics science, these are characterized like this. When highest dose soluble in less than 250 ml of water, Okay, it is called highly soluble. Okay. But not simply at 1 pH, you have to test it at the pHs in the range 1.0 to 7.5. That is the pH range normally you cannot cross in the genotype. And hence, by measuring the solubility at different pH ranges in between 1 and 7.5, basing upon the solubility, you can identify a drug as highly soluble or low soluble. Similarly, for classifying it as highly permeable, so it is based upon the fraction absorbed. Fraction absorbed is a kinetic parameter. Fraction absorbed is greater than 0.90, that is 90%. So fraction absorbed, how it is calculated is that means the drug is given by intravenous dose in which there is no absorption process. Directly the drug is placed in the vein and hence the SS, that is total availability 100%, and assessment of bioavailability is based upon the area under the curve in pharmacokinetics, and then administer the drug orally, and then again follow the blood level curve and determine the area under the curve. So area under this curve, oral route divided by area under the IV route, it gives F. F is a fraction, 0.9 lakh. If it is 1 means complete 100% absorption. But when drugs are given orally, you can never expect 100% absorption. It will be less than 1. In percentage, it is less than 100%. So such a figure is taken as a measure of permeability when it is greater than 0.90 F value, or in terms of percentage 90% is called highly permeable. Then so to assess whether it is highly permeable or not, permeability character, it is not enough to find out the p value, partition coefficient value. Partition coefficient value is one important contributor, but apart from that, the inherent permeability also depends upon the molecule size. In the partition coefficient determination, we have not taken into account the size of the molecule. But in the in vivo absorption, the size of the molecule also plays a role, and hence partition coefficient, it takes into account the water solubility and oil solubility. But that means lipophilicity and hydrophilicity it will consider, but not the size. Then it is not suitable parameter. More important parameter is permeability. And for knowing a permeability, you have to do an experimentation in animals. So initially you have to do the experiments in animals, and later on in the phase 0, the same thing you have to do in humans and confirm the character. So several modern organic drugs belong to this class 2 only and exhibit dissolution rate limited absorption and needs enhancement in the dissolution rate, and dissolution rate is directly proportional to solubility, and hence they require enhancement in the solubility and dissolution rate. And a 13th principle, dissolution rate is majorly influenced by solubility and particle size of surface area. There are several other factors also. If you recall the famous noise-Whitney equation, there are several other things. But all those other factors, they will be constant when you are doing the formulation work. And in the formulation work, the two factors that you can manipulate or manage are the solubility and the size, particle size, and otherwise the surface area. Both are inversely related. So these two characters can be modified, particle size and solubility. And the dissolution rate is given as, in simple sense, solubility into the surface area. So directly, the more the solubility, more is the rate. The more the surface area, the more is the rate. And surface area and particle size are inversely related, and hence the less the size, the more is the surface area, and hence the less the size, the more is the dissolution rate. So these two things are altered by several means in the formulation, and other factors remain constant, and several methods are developed based on these principles for enhancing the dissolution and absorption rates in the drug product development. And so many methods are available. And then techniques, we consider only a few industrially useful techniques. Several techniques are there, and not all the techniques are readily applied. But the techniques most widely used in commercial technologies are based upon like this various pharmaceutical principle is like this. One is the particle size reduction. So we have seen already the particle size reduction. If we reduce the particle size, it is known as the micronization down to 1 mu, 1 mm, up to 1 mm. So if you go to micron range, like 10 microns, 20 microns, okay, if not exactly 1 micron, okay, micronization, this is normally done by milling only. Okay, jet mills and used, or fluid energy mills are used in industrial large scale for micronization, and micronized form of drugs are also made available since long, and they are being used. The first application of biopharmaceuticals is in fact using the microfined products in the design of products like microfined as well. It's over the last 40 years it is still popular. Why 40? More than 50 years it is still popular. Okay, microfined means fine size. So in the beginning years of development of biopharmaceutical science, when a lot of things are discussed about micronization and its advantage, and shown with certain drugs commercially success, then one pharmacist shown great interest in this micronization and blood level pictures and try to use this technology to a drug called tolbutamide. One time it is the most popular antidiabetic drug. Even today also it is in usage, tolbutamide antidiabetic drug. And it is being used without fully knowing the consequences. The microfined tolbutamide is made into a tablet and put to use, and the doctor and the pharmacist, they don't know what exactly the result, and a drastic result is obtained. The patient went into coma. This is reported in the literature, medical literature. The patient went into coma because the micronization resulted in a very rapid absorption, and simultaneously the tolbutamide levels in blood plasma reached high, and equally rapidly the blood glucose levels depleted and got lower than the minimum requirement, and the patient went into coma. Okay. Then the medical journal identified knowledge is good, but the biopharmaceutics knowledge should be used with care, with caution and care and caution. So understand what is needed for the body. It's not that for each and every drug we should use this technology, but only when needed. And hence the more important thing is the therapeutic qualities needed. So in developing a process form or a technology to be used, etc., first we have to understand what is the therapeutic quality required in the product. What is the therapeutic requirement? So requirements are there for drug, particle size requirement, tablet-wise dissolution requirement, etc., disintegration requirement. In the same manner, okay, for therapeutic qualities also you have to understand what is the requirement. Do you require a very fast absorption and onset of action within 10 minutes, within 5 minutes you need or not? Okay. So if it is needed, then only you can go for micronization. micronization. So micronization is used in the cases where fast absorption is needed for treating acute disorders like your headache, aspirin. Certainly it's a good drug, but tolbutamide is used chronically. If there were a chronic disease, it need not be very fast absorbed, but steadily it should absorb, start producing reduction, then continue that reduction over a longer period of time. That is what it is. The type of therapeutic effect needed is that. And hence, in such cases, this type of technology is not at all needed. They identify. And hence, before putting any technology or any technique or any formulation, type of formulation, we should first thoroughly understand what is the therapeutic requirement. Accordingly, you have to resort to different techniques or technologies. And hence, several now micronizal forms are available and it is very successful. And simple technique also industrially feasible, milling. Several advancements have taken place in the equipment also. Modern machines are available, etc. And then an extension of this is the nanoparticles. So nanoparticle, you know that the size is still less in the nano range. In the nano range, it's wrongly written as 100 to 200 nanometers, not mu. It's nano, nm, not mu. So correct it, 100 to 200. Okay. And several drugs are now available in the nano form, that is nano suspensions they are made, nano suspension. Good examples in recent, these are the examples that of the drugs available in the market, like spironolactone, nano suspension is there in the form of course, tablet only, and phenofibrate is there, resiofalvin is there, progesterone is there, so such other drugs commercially now available in different trade names, and the nano suspension, the particles, nanoparticles, preparation is nanoparticle only, but normally they are available as suspension sometimes, or even sometimes they are made into tablets. So that means the technique recommended, those several techniques are reported in the literature for making nanoparticles and nano suspensions. The wet milling is one technique, and high-speed homogenization is another technique, and normally these two are combined. Wet milling or media milling with high-speed homogenization is the best technique many companies utilize in making a nano suspension. But after making a nano suspension, it is rarely that suspension is available, the preparation is made available as suspension, a typical suspension. Instead, it is spray dried or freeze dried. These are the two techniques used to convert into a powder form, solid form, spray dried and freeze dried. These techniques are joined at the end, and that is to make it into a tablet or capsule. That is the most convenient, comfortable dosage form for the manufacturer, for various purposes like transportation easy and stability will be more, and then for the patient also, administration of a tablet and capsule is as easy than the suspension liquid form. So all these commercial nano suspension formulations are now available as the tablets and capsules. But nano suspension, nanoparticles, in fact, the technology is not new. For pharmacy, it is a very old one. So we studied it since long. We are studying since long. Colloids, the concept of colloids. Very important chapter in physical pharmacy. Colloid, they are nothing but the colloidal particle. This category only. Their application is well recognized long back. Even though the nano delivery systems or nano suspension, these terms are not introduced at that time. So colloids are there, they offer the same advantage of rapid penetration, means rapid absorption today, and the one is the absorption is increased manyfold when compared to micronized form. The two reasons are there. The enhancement in absorption is due to solubility enhancement. In the case of nano, one factor is solubility enormously increases because of the reduced size, and another is the nanoparticles, they can cross the barrier. They can cross the barriers, whereas microparticles, they cannot cross the barriers. So these nano systems also, they can penetrate directly and enter, and the absorption and penetration is much higher in the case of nano systems. So these are the things that are. So now at the level of institution, this of course, these pieces of equipment are readily available in industry. The most advanced versions of these equipment they utilize. But in institutions, the difficulty is how best these can be done. And nano preparation, okay, nano preparation without having stabilization at the end. For stabilization, these are the two techniques. You quickly dry it by using either spray dryer or freeze dryer. Without that, there is no point in making a nano suspension because they will not be stable. Coalescence occurs immediately, and hence the advantage is lost. So these are some of the limitations in the academic research. You cannot go to certain extent using the advanced facility. But certainly the concepts we can apply and prove. This is in continuation. The another important technique is solid dispersion. Solid dispersion. Now, so I recollect my experience as a teacher. When I asked the student to do a PG student or a PhD student on solid dispersion, they show a reluctance. It's the solid dispersion is a very old technique, etc. But whether technique is a primitive or old, it's of use. It's of use. Even today, it is most widely used technique to handle insoluble drugs. Okay. So solid dispersion is a simple. It is simple technique only. You know that it is dispersing an insoluble drug, a molecular dispersion of insoluble drug in a water soluble carriers, and hence because the carrier is water soluble, really it creates problems. So you studied in the book and prepared in the lab, PVP dispersion, okay, HPMC dispersion, and you cannot go beyond a particular concentration because they are highly hygroscopic. You will not get the product. Though the dispersion occurred, but recovery is very difficult. Recovery is a mass only. Sometimes with the PVP or HPMC is a large proportion. Unless you use such a large proportion, it is not possible to get the required enhancement. But these problems can be avoided if you use industrial techniques. So a solid dispersion definition remains the same. Different types of solid dispersion are also known, and all involve a carrier molecule and an insoluble drug made by different techniques which you have studied is a common solvent method, a fusion method, etc. And in the laboratory scale. But in the industrial scale, this will be general principle remain the same. And the solid dispersions provide rapid dissolution because of enhancement in the solubility character, because of the presence of solubilizer. They are otherwise called solubilizers. PVP is a good solubilizer. PEG is a good solubilizer. So because of the solubilizing effect of these carriers, as well as when you dry from a molecular dispersion, it dries as the molecules. The particle size is reduced to molecular level, increasing the surface area enormously, and hence the dissolution rate also increases. And hence the same principle is applied, but the techniques are slightly different. The first technique industrially used is known as the spray dry dispersion. Spray drying is needed. Spray drying, which industrially can easily be applied. Okay, spray dried products. Several spray dried products are now made available, not only for drugs, for foods, etc. Spray dried form. Okay. Discuss the same formulation, you apply a spray dried, and this is known as spray dried discussion. Then another is hot melt extrusion technique. So this is known as HME. It is also a dispersion. So excipients used include HPMC, PVP, HPMC, stated succinate, and iodragids, soluble iodragids, L100, etc. So when they are made, so heated like all techniques, same, melt it while hot, extrude it, and then you get the solidified extrude and grind to size. So extrude size may be large, but inside the solidified extrude, you find a drug in the minuscule form, in the molecular sized form, and hence the resolution greatly enhances. So the principle remains the same. The size greatly reduces, that is the principle. And because the carriers are water soluble, the hydrophilicity will be increased and rapid wetting occurs, etc. And because in this process, when it is dispersed in a polymer, high molecular polymers like HPLC, PVP, the crystalline forms are converted into amorphous form. Like this, multiple reasons are there for enhanced resolution rate in the case of a solid dispersion, and it can be made into two forms: SDD, spray dry dispersion, and HME, hot melt extrusion, in the same type of materials, etc. And several drugs very widely used: ritonavir, and verapamil, and itraconazole. In fact, with itraconazole, for a long time there is a real problem. So even now several companies are still trying. It's so insoluble, and hence you are not getting the required dissolution in producing the generic product. One of the important characteristics is dissolution. They take the innovative product. Innovative product is wonderful, itraconazole, and even a very big established organization like that Reddy Lab, sometime back in late 80s, they find a real problem. They could not. So it so happened that one of the person inside, a manager level person, is known to me, and incidentally, in the academics, though we are not using spray dried and hot melt extrusion technique, several times I have given the topic of solid dispersion, enhancing the dissolution bioavailability formulation. One such drug selected long back is itraconazole. And I said, yes, we can easily get it by solid dispersion. First they didn't believe. Then I sent the tablets. And send the tablets. So first I send the data. Then again worrying whether this data is what is it. So then I send the tablet. I will send the tablet. You yourself can check the resolution in your own way in your lab. So that gentleman repeated the same tablet, sent the tablets and repeated, and the surprise is that dissolution is excellent and even far beyond the innovative product. Then he agreed that it is good, but how best we can put it in industry. Said that is your headache. There are ways and means. Probably by the time he is also not aware of the spray drying technique for solid dispersion, etc. And then industrial feasibility should be there. So there it said, okay, we'll going on. Concept is good, but we will try to take up this later. And so concepts are very important, itraconazole. And then he put an interesting question. So the tablet which we made is given a dissolution above, better than the innovative product. Then they worried whether it can be accepted or not. He said it is better and you can accept, but he says our bodies want a product similar to innovative product, a dissolution character similar to innovative product. So then, so later on, of course, now today there are techniques developed. We can combine several optimization techniques have come, and then later on, after some years, so I combined the optimization techniques, statistical optimization techniques in the preparation of tablets using solid dispersion and could achieve whatever resolution is required. We can now suggest. By the time we retired from that company, moved to some other company, and so it is not possible for us to discuss further. Okay, so it is. So later on, we'll see after some time about the generic products. They want to duplicate, but there is no point. In the case of IR tablets, immediate release tablets, the innovative product will give certain resolution. The developed product may not give less than that, but may give more than that, no problem. But in the case of other types, like controlled release products, yes, when they say this is the rate of release and this is the peak, the innovator is getting only this peak means the product which is the development is required should also give the same generic product should also give the same peak, not more than that. But I don't think any reason in the case of conventional tablets, it is immediate release, fast release only 80%. Instead of 80% in 10 minutes, if it is 85, 90, it's not affecting its efficacy. Okay. So this is the incidentally. So as the tablets and capsules, these can be easily dispensed, and hence, now what we have to remember is that whatever technique and technology is selected, ultimately it should be feasible for us to give tablet or capsule dosage form, preferably tablet dosage form, because technologically and marketing wise, tablet is the most convenient dosage form. Then we have the several other commercial techniques in this category. There is these techniques are suitable for class 2 as well as class 3 drugs. Class 3 means the permeability is a problem and how to tackle, how to improve. There are ways and means, and all these utilize the lipids. So lipid-based delivery systems. They are developed later on, lipid-based delivery systems like self-emulsifying drug delivery systems. Some are called SMEDS, SMEDDS, means self-emulsifying, self-micro-emulsifying, and another is self-nano-emulsifying. So essentially they are self-emulsifying, means when added to an aqueous system, they instantaneously emulsify. The advantage is that emulsified means the fine globules in the range of micron or nano, they are resulting, and these are lipids embodying the drug. Drug is embedded in this, and hence the lipid crosses the biological barrier. By doing so, it also encourages the lipid drug to cross the barrier, and hence rapid absorption is observed. So these are all in the same category. Materials are available and used commercially. Modern surfactants, fat lipids are there. Surfactants means normally non-ionic surfactants are preferred because they are non-toxic. They do not alter the permeability character, and many commercially available non-ionic surfactants have come, and solvents, co-solvents can be used easily. So these are ultimately these systems after preparation. This is only emulsification only, and so emulsifiers, after emulsifiers, and so the emulsification equipment only needed, and many advanced versions of these equipments are available commercially and easily can be done. And they are soft gelatin capsules. Finally, they are supplied in the form of soft gelatin capsules. That's why they are more costly rather than tablets. So still difficulties are there for converting into a tablet dosage form or a have hard capsules, but all are available as soft gelatin capsules. And the same technique is resorted to long back, since long. These are, of course, developed during the last 20, 25 years. But even earlier, 40 years ago, also use of the co-solvents or solubilizers, particularly lipid-based solubilizers, is there to solubilize, and the solution, because it is non-aqueous, because it is aqueous solution, water-miscible solvents, and hence they are made into soft capsules. Soft capsules bases. So one such drug is nifedipine. Nifedipine. So nifedipine is highly insoluble. Highly insoluble. You will never get in the solid dosage form like a tablet. Okay. The desired concentration. Okay. It is available as soft capsules. It is dissolved in non-aqueous solubilizers like non-ionic surfactants. They are highly soluble. They dissolve several organic compounds, and this solution is enclosed in soft gelatin capsule. At that time, okay, that oil, after rupture of the soft capsule, the oil contained medicament will be released, and in the system, in the body itself, there are what are known as the bile salts. And bile salts encourage the emulsification of the oils in the state or even the fatty food materials. They are emulsified by the bile salts, and hence inherently the system is having the required emulsification mechanism. But later on, to be made applicable for variety of materials, etc. So self-emulsifying systems for drug delivery. So in the formulation itself, we will be providing a more powerful emulsifying agent along with the lipids required to form either a microemulsion or an nanoemulsion. The result is the rapid absorption. So drugs like testosterone, calcitrate, cyclosporin, valproic acid, and such other compounds are today available in the form of soft capsules using the techniques self-emulsifying drug delivery systems. And hence, this is another industrially useful technique based upon the same principle, bioconjugate concepts only. And then another technique, interesting technique developed is inclusion complexes with beta cycloaddiction and such other cycloadditions. Among all cycloadditions, beta cycloaddiction is more popular. It forms inclusion complexes. It has a cup-shaped structure. Inside it is more lipophilic, and hence the lipophilic insoluble drug will be entered into inside the CD. The dimensions are very low, molecular dimension, and outside it is hydrophilic, and hence mix well with water and form micelle-like structures. Micelles, surfactants, they form the same way. Micelles, in the same way, they also resemble micelles in water. And drug is embedded in the CD and is molecularly dispersed. And these cycloaddiction complexes, they are reported as having several pharmaceutical benefits like enhanced solubility, absorption, stability, okay, suitable some even for control release, etc. So many applications are reported, and for enhancing, since long, thousands of articles you will find in the academics, journals, etc. These CD articles, circulation work, but several years the industry has not touched this. The reason is the cycloaddictions are very costly earlier, not now. Earlier they are very costly because it is made initially by chemical synthesis or chemical processing only, which is found to be very costly affair, and when the cost of excipient is more, normally they do not find a place in formulation. The excipient should serve the purpose, but at the same time it should be cost benefiting, cost effective. And in later 90s. A fermentative means today you call it as a biotechnology process, microbial conversion process is developed for making the cyclodextrin. With the result, the cost of the cyclodextrin has come down enormously in starting from 2000 and such years. So then started several companies where it is known a wonderful molecule, but because of cost nobody touched it. Then almost several companies have made use of the cyclodextrin. Now you find several formulations contain the beta C. It is only an excipient, and sometimes as a marketing gimmick, the certain manufacturers they write the CD on the label also beta CD. They need not, but if they write also there is no objection. And some doctor friends inquired me what is the CD, sir, what drug it is. I have not known earlier. But I said it's not drug, sir. It is only an excipient. It is there inside to enhance the dissolution, absorption, etc. And hence the performance will be definitely. So almost initial stages, simple drugs, but creating problems like pyroxicam. Pyroxicam CD is first very old application of CD in formulations. Pyroxicam CD formulations are there. Nimsalide CD formulations are there. Of course, later on the nimsalide drug itself is banned. So like this, for some technologies, cost may be a barrier, but provided the cost effective such a excipient selection using the techniques, this comes under the technique of complexation. Complexation is a well-known technique, has a lot of applications in pharmacy, and hence in formulation also, particularly the cyclodextrin complexations find wide application today, and the complex form, we need not prepare complexes separately, even when they are mixed with the drug and in a certain proportion. When formulated as a tablet, then the tablet dissolves or suspends in water, then the complexes will form and they help in enhancing the process of absorption and dissolution. The other methods used also in industry, but to a less extent, like use of surfactants. In tablets and capsules, surfactants can be used, non-ionics like phenes and poloxamas, glucide, polysorbates. So all are like these compounds. They are mixed in sufficient in some concentration. The tablet mixed and encapsulated, and the surfactants they enhance the solubility. They are very good solubilizers. Okay, by forming micelles in the presence of water, and surfactants also lower the interfacial tension and hence they make the hydrophobic drug more easily wetted with the aqueous fluids that itself will enhance the dissolution. And when it comes to dissolution and formulation, it's not exactly one factor is responsible. Several factors collectively they enhance the dissolution rate. Individually, it may not be possible to identify its effect. Say, for example, the lipophilicity. Lipophilicity is always detrimental, and the tablet surface should be hydrophilic. If it is hydrophilic, definitely the dissolution will be fast. And hence, how to create hydrophilic? So, water soluble excipients only. We will use of lactose. That is the reason why lactose is more popular. We convert the tablet hydrophilic, and hence dissolution will be more. So, if you use lactose, that is one factor. Along with the surfactant also enhance the rapidity with which the surface is wetted, and also it increases the solubility. So combining all these factors greatly enhance the dissolution rate, though individually a significant dissolution enhancement may not be observed. And that is the reason why The surfactants when used in formulations, definitely they enhance the dissolution rate. And so that it is easy to use, mix them with other excipients and use it. Soluble agents also in the same manner, PEGs, PVP, non-ionic surfactant, Soluplus, a wonderful molecule nowadays, find wide application in solid dosage forms. And these are used in very small proportions, otherwise the difficulty in tableting and difficulty in getting good flow, these technical problems will come, but they are very much useful and to get the good dissolution from a tableted system. And use of salts, amorphous forms, metastable polymers, these are all the techniques recorded in the literature and still it is used. And in fact, this is a subject ever demand and going on. And for example, you know that polymers, certain polymers, okay, polymorphic form. So if you establish a new polymorphic form that is reasonably stable, metastable form, and that itself contribute and it is considered as a new drug. And so it is, you should prove that in some way it's having more therapeutic effect. So certain polymorphic forms, they exhibit better absorption or distribution character with the result they are preferred over others. Like that, the say, drug may be converted into amorphous form, and amorphous forms, if you make it available, certainly they can form a good means to enhance the dissolution bioavailability. Like these appear to be simple, but at the same time, they are very much useful in formulations, and you can decide. And then solvent deposition on inert carriers. It's similar to solid dispersion, but difference is here these inert carriers. Like inert carriers, even in the tablet and capsule manufacturing, you are using excipients, a number of excipients. These excipients are all inert only, and they are tolerated in tableting. Whereas the carriers used in solid dispersion, HBMC and PVP, you cannot add in a tablet. Tablet will become too hard. But the tablet excipients used, okay, like fillers, microcrystalline cellulose, they are anyway compatible and tolerated in tablets to any extent, reasonably. And then the drug is deposited over such excipients in minuscule form. It is called a solvent deposition, and they are very much suitable, easy to manipulate. The drug is dissolved in a solvent and sprayed over the tablet mix over the excipients and mix it thoroughly. And then during this, when solvent evaporates, because it's already in solution form sprayed, and while mixing the solvent evaporate, leaving drug in minuscule form deposited on the site. This is always known as solvent deposition. This is a simple technique used, and several carriers. These carriers are not different from the excipients used in tablets, and hence can easily be manipulated into tablets and capsules, but with great enhancement in the dissolution rate. Like this, several simple as well as commercial techniques that are based upon the concept of the biopharmaceutics, essentially making use of the particle size manipulations, solubility manipulations, and enhancing the lipid character, etc. And several such things, advancements are made. And then in the formulation and in biopharmaceutics, you come across many times with the dissolution and dissolution test. We've seen that almost all pharmaceuticals include dissolution rate tests for all the solid dosage because it has several applications in this area, drug development process. As a QC test, works as a quality control test over all the solid dosage forms, and in the finished product testing, it is used, and sometimes to in the in-process quality control also, in critical cases, you can subject to this test, and to ensure batch-to-batch uniformity. This is very essential. Batch-to-batch uniformity. So earlier it is only considered as the content. Many times the content uniformity. But now the more important is, though the batches contain the same amount, same drug, there is no guarantee that in the dissolution character it is uniform. So batch-to-batch assessment is made based on the dissolution rate. And as a research tool, secondly, in the formulation work and other research works, to evaluate the effect of variables. Evaluate the effect of variables initially. The dissolution is a good tool to check the effects and to find out the best. So for R&D purposes, it is widely used in formulation development. And similarly, to predict the in vivo performance of drug products. So it is not that every time the in vivo evaluation, either the blood level comparisons or the therapeutic pharmacological evaluations cannot be done on each and every batch. There are many times it cannot be done because of the limitations, not needed regulation wise also. And in certain cases, an in vitro measure is this dissolution rate. So dissolution rate test is taken as a measure to predict the in vivo performance. But this prediction is possible only when one establishes the in vitro in vivo correlation. And hence for such in vitro in vivo correlation, you need to develop a good dissolution test, and of course in vivo study has to be conducted appropriately. And so in vitro, it is a in vitro measure of bioavailability. So the term bioavailability is always means evaluation in human subjects only ultimately. But as a prerequisite before going to bioequivalence study, you can make comparisons in the in vitro behavior. That is in vitro measure of bioavailability, especially in the development of generic drug products. Always a comparison with the innovator product is needed, and several alternatives will be taken up, and these several alternatives need not be subjected to bioequivalence test, but only the final product will be subjected, and before that you need a comparison. For that, many times the in vitro dissolution test is used. In vitro dissolution performance of all the alternative products is compared with the innovator product to select to select the best. Like this, it has several applications. But the dissolution rate test development is an important topic in biopharmaceutics, which is very much needed for drug development research at different stages, and the conditions to simulate. That means whatever test we do, it should simulate the in vivo condition. And first thing is a body temperature. You know that 37 degrees. Then GI tract movements, peristaltic movement will be there. The contents will be continuously subject to mixing, and that mixing also affect the absorption process and hence the blood levels achieved, etc. So you have to simulate this movement of depth by adequate mixing with a stirrer, and here the different types of stirrers and different speeds are possible to use, and even the pharmacopoeia recommends a variety of stirrers. They are known as apparatus 1, apparatus 2, like that. Okay, and some paddle stirrer, sometimes and basket stirrer. Okay, different manipulations of that, and as many as 8 different apparatus are designed or are made official in all the pharmacopoeias. And with each type, there should be a provision to alter the speed more precisely, Speed here means not heavy mixing, it's only at a low level only. Like you find in the pharmacopoeia, speeds ranging 50, 75, 100 only. Not like 500,000. The speeds which we will be using for preparation of emulsion and suspension is not that type of mixing. It's only low speed only, but it should be precise in the sense that the same 50 RPM should be obtained throughout the study. The study may be 2 hours, study may be 10 hours. Whatever it is the length of the study, the same rotation type of rotation RPM should be maintained. That's very important. And hence, the design of the resolution rate test equipment is a very critical one. More precise technology should be used in designing this motor and the spindle, etc. That is the reason why the resolution rate apparatus is very costly. Otherwise, it is only simple starter attached to a motor, and other things are only a 1 liter jar, a water bath, simple assembly, but precision is more important. Precision means the repeatability, the same type of result we expect when it is used repeatedly or continuously. So that's why to design these precise motors, the cost is enormously increased. Then the sink condition has to be maintained. So in the biopharmaceutics, the sink condition prevails during the absorption phase, okay, and it is the concentration gradient that is the driving force in the positive diffusion. You have seen the first principle itself is positive diffusion is the major mechanism. In the positive diffusion, the driving force is the concentration gradient, and the difference in the concentration of the drug in the gut, GI tract, fluids, and the plasma, other side of the membrane. So, large concentration gradient is otherwise known as sink condition. That is the driving force. And what is happening in the system is the concentration in the gut is several times higher than the concentration in the plasma. Otherwise, the plasma concentration is very, very low when compared to gut concentration. Gut concentration depends on the dose, and hence doses are large amounts are given. Sometimes 100 mg itself is low, but many drugs 400 to 500 dose is given, a large amount is given, concentration. The fluids in the system is maximum 100 to 120 ml in the stomach. So concentration is very high. Whereas when once the drug absorbs into the plasma, it's not staying there only. It is being distributed over a larger area and enter into various tissues and organs. With the result, the concentration in the plasma is very small. So plasma concentrations, depending upon the dose you find in micrograms per ml. If the dose itself is 20 to 50 mg, the concentration will be fraction of a microgram or nanogram. Still, small dose means it is still fraction of a nanogram or a picogram, etc. So concentration in plasma known as CP is very, very less when compared to CG, which is very large. This concentration gradient you have to represent in the in vitro system. How to represent in vitro system is, in the in vitro system, the concentration gradient for dissolution is the CS minus CT. CS is the saturation concentration of the solubility of the drug in the fluid. CT is concentration generated at any time. Okay, because at different times you will be taking sample, so concentration will be varying. So at any time T, CT is the concentration. And hence the concentration gradient is CS minus CT. This should be large. And how large it should be maintained? CS should be at least 10 times greater than CT. 10 times greater than CT. And hence, now how to select the fluid? The fluid is selected in such a way, the dissolution fluid you have to select. Okay. And selection of the fluid in such a way that you will select a fluid in which the drug is having high solubility. CS is high. You know that the solubility is different in different solvents, but solvents cannot be used, it's only aqueous system and have buffers of different pHs. The general rule is if the the drug is acidic, alkaline fluid circuit, because in which the compounds dissolve and CS will be happening. And basic drugs, the acidic pH, reverse, vice versa. Okay. And for neutral drugs, so pH has not much influence on the solubility. In such a case, you can add other materials like surfactants. You know that surfactants enhance the solubility. So officially also it is accepted. In pharmacopoeia also for several drugs, the surfactant solution, either surfactant added to water at a concentration sometimes as high as 10%, sometimes 1%, 2%, and sometimes a surfactant added to a buffer is selected as the fluid for neutral drugs. And if the drug itself is water soluble, then simply water can be used. Lithium is highly water soluble. So in the pharmacopoeia, you find water only as the fluid. And like this, the selection of the fluid on what basis means, which is the basis is you have to select a fluid in which the compound is having high solubility and at least CS means solubility or saturation concentration is 10 times more than 10 times than and then the concentration that is generated. So you can, depending upon the dose, the amount in the tablet and volume of the fluid taken, you can calculate what is the maximum concentration CT developed when it completely dissolves. Then multiply with the 10, that should be the minimum CS. So you select a fluid in which the compound, said compound will be having so much solubility, that would be one ideal way. Then how much fluid, how much selecting fluid is enough. Then large volume is recommended. In all the pharmacopoeial methods, 900. But you see that in the body, you never find 900 ml anywhere in the either in the stomach or intestine. Even then, 900 ml fluid is recommended, large volume. This is because if the volume is large and the CT will become less. That means the two ways that to maintain sink condition is either increasing CS or decreasing CT. So one way is decreasing CT. That means concentration of the dissolved drug. If you take large volume, because dissolution test is a closed test, a closed compartment test, the vessel, all the 900 fluid will be there only till the test is over. At the most, you can take a sample of 5 ml and replace it like that. Okay, and hence the CT, to keep CT low, to view the sink condition, 900 ml is recommended. And like this, you have the body, like these are the conditions all you have to maintain. Now, which way you have to finalize? So anything official, if you want to do official test for quality control purpose, it is okay. But what is the major objective of dissolution rate test is to make it as an alternative to bioavailability test, especially for development of generic formulations, etc. And in which case, a dissolution rate test developer should be meaningful and should give the in vivo performance, bioavailability performance, and it is possible only when you have established a good correlation between in vitro and in vivo. So, and any change in the above conditions is allowed. Not in the pharmacopoeial test. For pharmacopoeial test, quality control, exactly you have to follow the same. That is for quality control purpose only, either to accept the sample, accept the batch or not. But for other things, developmental purpose, an in vitro dissolution rate test should first be correlated well with the in vivo. So that uses the concept of in vitro correlation. Suppose you basically set one methodology and then the in vivo experiments, take some products, 4 products, perform the in vivo plasma concentration data and arrive at some parameters and try to match with the dissolution parameter. You may not get. Then think about which way you can alter. So sometimes the alteration will be one. You cannot go on altering the temperature. That should get the body temperature. Okay. But what you can alter is, say for example, you can alter the agitation, speed, adjustments you can make. Okay, another trial. Or alternatively, you can alter the fluid composition Okay, make it more sensitive or less sensitive by altering the composition, etc. And hence by such manipulations, you carry out again the revised resolution and again make a correlation and now select the best conditions which give the best correlation with the in vivo data. When once you finalize the set of conditions to give a good in vitro in vivo correlation, later on you should not change those conditions. If you change those conditions, the correlation is lost and you cannot take it as a predictive measure. And hence, the aim of this development, resolution rate-based development, is to get a good in vitro in vivo correlation. That is one thing. And then a test developer should be able to discriminate. There is another objective. It should be able to discriminate the little differences in the formulation. For example, take a factor like the speed. Normally, why the pharmacopoeia and other test results use only 50 and 100? If you use 500 RPM, intensive mixing will be there. With the result, the little difference in the dissolution character between two dosage forms is nullified because of the high mixing, and hence that is not suitable. Mixing should always, in the dosage itself, you fix a range 25, 50, like that, 75. You check where you will get a good correlation, you can fix it like that. And hence, the another objective apart from in vitro in vivo correlation, a test should be discriminative. It should serve the purpose of distinguishing two formulations with when they have a little difference. That is all. Then, like this, this is a good area of research in biopharmaceuticals. And then the bioavailability. It is so it's simple. Okay, the measurement of both rate and extent of absorption is otherwise called bioavailability, and an important that bioavailable dose means the bioavailable dose amount absorbed by the administered dose is otherwise known as the bioavailable or fraction absorbed. And for this calculation also, okay, F fraction absorbed is defined like this, which is very much needed in the pharmacokinetic analysis of data. And then bioavailable fraction F is normally estimated by comparing the oral route against the IV route, where IV means 100%. And hence, by conducting a test two times, one giving the dose intravenously and another giving the drug orally. And this is absolute bioavailability. Okay, the same one. And in this graph, the absolute bioavailability, this is a case study with nimodipine. Nimodipine is another drug, of course, old drug, but is a very typically insoluble drug. That's why the oral bioavailability is only 1.17%. Okay, fraction. So you see the lowest curve is the oral, and the top curve is the intravenous. Intravenous is the top curve. Okay, because there directly the drug is placed inside the vein, so starting point is at the top and gradually decreases. And in between you have another route like nasal. Nasal route found ideal because sufficient absorption without much deviation. Deviations are also important. So in the case of IV, you see that initially concentrations are very high, but later on, after some time only, okay, it is stabilized. So instead, in the case of nasal also, you find a relatively rapid absorption, higher absorption compared to oral, and the curve is a gradual one. Like the route, for selecting the route, this type of assessment is done in the clinic in the first animals, later on in the phase 0, sometimes in phase 1 also the studies are made for selecting the route as well as for fixing the dose. If you alter the dose, the curve, shape of the curve will be more. And anyway, you will be finding in the animal studies and correlating in the zero phase with the concentrations, etc., you find out what is the minimum effective concentration and minimum toxic. They are known as the therapeutic range. That is an important requirement that should be established, ranges, and then to select the route and dose, the criteria is that the concentration should reach within that range as early as possible and maintain over a longer period in that range, basing upon the elimination character. So such studies are made. Then this is the relative bioavailability. Is the oral route is compared with oral and the drug product and the standard. So compare the bioavailability of a formulation with certain other drug. Normally nowadays these comparisons are needed in the generic product development. So comparison, the standard is taken as the innovator product, and the formulated one, generic product is the test product, and hence the test product area under the curve by standard innovator product area under the curve is otherwise known as a relative bioavailability. And requirements of bioavailability and bioequivalent studies. So when we have to conduct bioavailability and bioequivalent studies, and this is for IND and NDA, investigational new drug application and new drug applications. When you are applying it normally, you need that bioequivalent study has to be carried out. And in the ANDA also later on it will come. So to establish the equivalence between, not only for final submission, but before that also to establish certain equivalence. That means early and late clinical trial formulation. So that means clinical trial formulations over a long period, and initially some trial formulations will be conducted, and later on refinements will be made, and then again you need to compare the bioavailability and bioequivalence for making such comparisons. Then similarly, formulations used in clinical trials and stability studies, so clinical trials formulation. Then finished final formulation will be something different. It is called to be marketed product. So you need the comparison of clinical trial formulation and to be marketed drug product finally. You have to prove that it is not changed in the final product. Similarly, you have to compare the stability during stability studies. So after subjecting to stability studies, the formulation used in clinical trials and the formulation used in stability studies, both are the same or not, you have to get the data and prove they are the same. So any other compound changes, okay, any other changes made. So changes in the components, composition, and manufacturing procedures, products, such changes are made. Whenever such change is made, you have to necessarily carry out again a bioavailability and bioequivalence study and submit the data for approval. And of course, ANDA for generic drug product development compares approval. And for generic drug, the company or the sponsor need not do all the developmental steps. Only they can formulate a product which is equivalent to the innovator product. But at the end, apart from other in vitro evaluations, finally, and support basis should also be now, rationale should also be submitted. That's why even the formulation techniques, there should be some rationale, some QBD, quality by design approach, or optimization approach, something like rationality should be shown today. It's not enough. A randomly written formula is not suitable. So a formula should be developed on rational basis. Evidence should be created, even for so simple things that you have added, say for example, a surfactant, a few mg, 5 mg of surfactant going for 500 and 200 mg dose. You have to substantiate why you have selected this. You have to gather the information. Okay, what is the benefit of adding this? If you already reported evidence is there, and you create, otherwise you have to create your own evidence and substantiate the inclusion. Like changes in the A&D means generic drug approvals, you need to submit at the end bioavailability and bioequivalence. Other like developmental studies and the other preclinical animal experimentation, phase 1 to phase 3, phase 4 studies, they are all not at all needed for applying for generic product approval. Only a clinical study need is bioavailability and bioequivalence. This is also a clinical study. And a change in dosage form like capsule and tablet, tablet, capsule, any change like that is made means necessarily also you have to get again the bioequivalence study data and submit and get the approval. This is an example of the formulation A and B2 formulations. Plasma level curve, the therapeutic range is there, and now two curves, formulation B upper one and the formulation A. You see easily that the difference, the products are the same dose, but in one case, the formulation A, more peak is observed. The peak is higher than the MTC. Normally, it's not advisable. Unwanted toxic effects may precipitate, and the concentrations decline fast because of the high concentration. The decline is also fast because always the decline elimination is rate of elimination is proportional to the plasma concentration. The more the plasma concentration, the more rapid will be the decline. So rapidly declared, and hence MEC is there, minimum effective concentration. In about less than 2 hours, the effect is lost. Whereas in formulation B, so steadily it increased, concentrations are all within the range only, and up to nearly 4 hours, 3.5 hours, it is in the MEC level. Like that, the differences can easily be inferred. So here the test is the plasma concentration means the bioavailability test only, and the inferences can be made and from these type of curves, you can calculate all the pharmacokinetic parameters, and particularly for bioavailability, it is comparing the two products, means here the relative bioavailability of the calculate, and based upon the area concept. Now this is bioequivalence. So when two or more products produce the same blood level curve, now they are said to be bioequivalent. Okay, same curves. Okay, that means both the rate and extent of absorption should be the equal, and so statistically the difference should not be significant. A little difference is allowed, but not statistically significant. So every time at the end, a statistical comparison has to be made on the parameters to be compared. For bioequivalence comparisons, you need to compare sometimes the Cmax and the area under the curve. So there should not be any statistical difference, significant difference. In the absence of tests, a 20% difference is allowed. The difference should not be, the parameter should not be more than 20%. And therapeutic equivalence means ultimately